I decided what would maybe be the best use of our time is just keep going with the whole process of cellular respiration. So I put a PowerPoint in for aerobic respiration, and it's just the, as you saw in the POGL, it's the link step and the Krebs cycle. So we'll talk about those two pieces, and then Friday we'll hit electron transport and chemiosmosis. And I think as far as the AP world goes, you see chemiosmosis every year. So that is a concept you definitely want to be familiar with. So we're going to jump into stages 2 and 3. Stage 1 was glycolysis. You guys should be able to map that out. So I really wanted a wiser me, but I didn't find one activity for that. But that's something you guys can practice on your own. If we were in class, I usually give you some green and red M&Ms and then make you walk through it, keeping track of the carbon atoms, hydrogen, and phosphorus or phosphates. That's something you guys could do on your own, right? You can draw that out, you can manipulate it out. I might have you do a video of that next week of you doing a manipulation. So after this, I have a POGL for you. Those of you who are in the Zoom today. But anyways, if you're here today, I don't need your POGL until tomorrow. I know some people were going to take advantage and sleep in today, in which case they'll be doing this on their own, and I'll need their POGL today. So getting into our process, you're going to do a POGL later. Glycolysis, we took a 6-carbon glucose and we broke it into 2 3-carbon pyruvates. So throughout this whole process, I think it's important to keep track of your carbon, right? Conservation of matter. And that worm lab that we were looking at, that gets into the, I like that because it gets into our first and second laws of thermodynamics about energy neither being created nor destroyed, only changed in form. And the same thing with mass. And what you find out when you measure the amount of mass lost from the food they eat and versus the amount of mass they gained, it's not equal. They've gained less than what they've eaten. So I think that that's really interesting, and it makes a good connection to entropy. Also, when you put them in a group together, you can measure the temperature change. And so there again, that loss of usable energy in the form of heat, which is directly related to cellular respiration. So that's a good concept to keep in mind along the way. When you break these bonds, you're losing some usable energy. The more bonds there are, remember, the more energy there is. So a 6-carbon, I know I've had questions about this on my test. Which one of these two have more energy? And I asked you guys that on Monday, and you were able to tell me glucose would have more energy than 2 pyruvates. Even though they have the same amount of carbon, there's more bonds in the glucose than there is in the pyruvate. Okay, so that's like just kind of recap of last week or of Monday. So every bond has energy, which means pyruvate hasn't released all of the energy that it has. And that's where the next steps come into play, is getting the rest of the energy out of that original glucose molecule. So we have to continue to strip off carbon, breaking bonds. Remember, as we lose electrons, we're oxidizing molecules. So we need to continue to oxidize the pyruvate. So our last slide on Monday, we had pyruvate, and it was going to go into one of 2 pathways. It was either going to go anaerobically through fermentation, which was 8-3 that I skipped, or it was going to go through aerobic respiration with oxygen, which was 8-4. And that's what you guys read last we talked. And you guys are taking the most beautiful notes that I've seen, so kudos to you. So if oxygen isn't available, we're going to go into the mitochondria. So remember, glycolysis was a very ancient pathway, happened out in the cytoplasm or cytosol. All organisms, no matter what, all organisms are going through glycolysis to at least release that little bit of energy that they can from breaking glucose in half, basically. Okay, but not all organisms have mitochondria. So now this is going to happen really just with our eukaryotes, right? Those that have internal organelles. So if oxygen is available, that pyruvate that we just made is going to enter the mitochondria where we can continue to break it down. And just a pattern to notice, every carbon you break off becomes CO2. So glucose, we've conserved all our carbon. We still have 6 at 2-pyruvate. As we strip that pyruvate one by one, carbon comes off and it's going to become carbon dioxide and NADH. And I'll show you that in a minute. So we need lots and lots of enzymes in order to go through the Krebs cycle. There's many steps and each has a intermediate, right? So every one of those steps requires enzymes. So again, not all organisms may go through this simply because they're too simple and they don't have the enzymes available. Like yeast, that little cellular, like single-celled fungus, they can go through fermentation simply because they don't have the enzymes needed to go through the Krebs cycle. Okay, different yeasts, different processes. So our pyruvate needs to be broken down into CO2. Just a review. Oh, hey, I went through and I was checking on my other letters. I missed these. Just a review of the structure of the mitochondria, this little peanut-looking thing. Remember, it has a double membrane layer. So the outer membrane, you literally call it the outer membrane. The inner membrane we can call the cristae. That's right here, cristae. And the filler of the inner membrane is the matrix. So inside of this space, we're going to need to know this. This is the matrix. In between the two membranes, you refer to the intermembrane. Inter is between. Intermembrane space out here, okay? So with our hydrogen ions, we're going to be moving them in and out. So it's important that we understand where those compartments are, okay? Oh, this is showing you some little mitochondrial DNA to remember that it's related to the bacteria. It has its own single circular DNA that replicates through binary fission, much like a bacteria, which is the part of the pieces of evidence that we have for the endosymbiotic relationship. What else? Looks like that's about everything. Remember the purpose of the folding. This is another thing. Remember, AP Biology likes to talk about the structure meets function. So the fact that it has all this folded membrane allows it to do more chemical reactions. So any membranes that are folded is increasing the efficiency, basically, of that structure. And that's what it would look like under an electron microscope. You can see all the inner folds really well. Cells that need more energy are going to have more mitochondria. So like your muscle cells, for example, need a lot of energy, so they're going to have a lot of mitochondria. So here you can see the inner folds, the cristae. You can see the outer structure. Looks like we've zeroed in here on the inner membrane, right? So that's where this is occurring. So we could talk about the outer membrane or the inner membrane. This should look very much like the electron transport chain that you saw in photosynthesis. It's very similar. It also uses cytochrome C. So you remember cytochrome was one of those inner membrane proteins that we used. So just referring back to that evolution piece that I have a tendency to talk ahead of my slides sometimes. But we talked about that endosymbiosis at the beginning of the year, okay? So membrane-bound proteins, remember I told you you should be able to talk about the structure of a membrane. So continue, like don't leave a chapter in the past. Try to make your connections and things that I've told you, you are likely to see in the future, right? So you are likely to continue talking about membrane structure and proteins being pumps or transport proteins or glycoproteins for communications, receptor proteins for signal transduction, things like that. Okay, so here we have a bunch of embedded proteins, and I can see NADH and FADH2 are going to play a role in this. What they're doing is these molecules are providing the energy for these hydrogen ion pumps. So just like we did with the electron transport in photosynthesis, you have hydrogen ion pumps. Electron carriers are going to donate electron and energy to these pumps where they're going to move hydrogen against their gradient. Okay, we need that gradient in order for ATP synthesis to occur. This is where the majority of our energy is taking place. So what we have to do before this is we have to make that NADH and the FADH2, right? We don't have those yet, so we need to make those. So that's what these steps are going to do. So here we have our pyruvate. This is our link reaction you read about in the POGL. So each of your pyruvates, so 2 times, so each of your pyruvates are going to be broken down through steps into acetyl CoA, coenzyme A. So remember, enzymes, substrates, active sites. The substrate doesn't always fit the active site. You need that extra piece, which is a coenzyme, in order to aid it, allow it to fit there. Every time we break off a carbon, we get CO2 and a hydrogen that gets put on NADH right there. That's our electron carrier. And if you think about a glucose molecule, if you could picture the structure, this is where I would be drawing it out on a board. Picture the structure, C, right? And then OH and an H. I need to learn to use the whiteboard. So for every carbon you have in glucose, remember it's carbohydrate, so each of them has a water molecule attached to it, right? Basically, OH and H. So it would make sense when you break that carbon, here's your oxygen from that OH, and here's your hydrogen that was connected by itself. So it would make sense that every time we break off a carbon, we get CO2 and hydrogen, which binds to NAD. So nicotinamine adenine dinucleotide. All right, so 3 steps. You get, for each of your pyruvates, you're gonna get 1 CO2, so a total of 2. This is our link reaction. You get 2 NADHs, 1 from each, and 2 acetyl CoAs. I need that acetyl CoA for the Krebs cycle. So where is this happening? Notice this is the outer membrane and the inner membrane. So this is really occurring like as it's transferring through the membranes of the mitochondria into the inner membrane, into the matrix. All right. And then depending on conditions, this step here has different levels of efficiency. So sometimes you'll see, like depending on the resource, they'll talk about 40, up to 40 ATP being produced, and others will say 32, 36, 34, and this is one of those steps that can make a difference. So depending on the efficiency of this transfer, you'll make different amounts of ATP, but we'll generalize that we get 2 out of that. So here we go. So this carbon dioxide, every carbon dioxide we make is going to be released, right, through breathing. Difference between ventilation, which is breathing, and cellular respiration, which is what we're talking about now at the cellular level. So you can see here the pyruvate, you made it in glycolysis. Know the beginnings and the ends of each of these individual processes, okay? Maybe not all of the individual steps, but what goes in and what comes out. So pyruvate enters into the mitochondria, and along the way, it's going to be converted into acetyl CoA simply by, well, really not simply, but for our sake, right? We're simply taking off a carbon dioxide and a hydrogen. So CO2 and hydrogen up here, NAD plus gets converted into NADH. Now remember, glycolysis needs NAD plus, so there's gonna have to be a way to recycle the NADH, and that's what we'll talk about on Friday. So pyruvate gets converted, that's your link step, into coenzyme A. Acetyl CoA is what enters the Krebs cycle. So I can see I had 3 carbons and now I have 2 carbons. Where did the carbon go? That's going to be a question in your POGL today. I took out the repetitives. I only asked you at once. It's going to go into CO2. So this is oxidation because you are stripping off electrons, stripping off hydrogen. Lots of things happening here. So NAD is an electron carrier. It's like putting electrons in the bank. named after Hans Krebs back in 1937. So he was like 37 years old. So this is also called the citric acid cycle, the next step. We're in phase 3 now. It's called the citric acid cycle because the first stable intermediate is citric acid or citrate. So that's how it got its name. So this part is happening in the inner space, right? So in the matrix, inside of the cristae, in the filler of the mitochondria. So many, many steps, 8 steps you see. Every step requires an enzyme. Every step will create a intermediate that is used for the next enzyme. So anything that affects an enzyme is going to affect the overall reaction, right? If any enzyme has a genetic mutation, it's going to affect everything that happens after that step. If temperatures are increased, if temperatures are decreased, you've already talked about what affects enzymes, right? So you could talk about this. Remember, they like to throw a wrench in the system on FRQs. They'll give you a situation and then they want to know, what if this doesn't work properly? So you need to be able to talk about that. Okay, so step-by-step catabolism, we're going to break down our acetyl CoA. We're going to make a citrate molecule. Because this is more complicated and happens within an organelle, it makes sense that it is a younger process. It doesn't date all the way back, right? So glycolysis, we can see in the bacteria that were here 3.5 billion years ago, forever ago, right? Oxygen started showing up in the atmosphere about 2.7 billion years ago. Remember, cyanobacteria makes oxygen. And so the atmosphere and the environment started changing, which allowed for evolution, organisms better suited for our environments, yada, yada. So our eukaryotes appeared about 1.5 billion years ago, and that's where we start having aerobic respiration within the mitochondria. So just talking about changes over time. So here's our 3 pyruvates, glycolysis, the link step, acetyl CoA enters. This is our recycling piece. So this should look very familiar to you, should look similar to the Calvin cycle. In this case, CO2 is going in, combining with a 4-carbon molecule to form citrate. So that is why it's called the citric acid. The first stable intermediate is citrate. Because you have 1 pyruvate going in per cycle, and glucose has 2 pyruvates, it's actually going to have to go through the cycle twice in order to break down your entire glucose molecule. Okay, so this is showing you a lot of things happening. So we can see the 6-carbon citrate. Notice the reds. This is your original carbon. So this is your glucose carbon. I said CO2 going in because I was talking about the Calvin cycle. Acetyl CoA is our 2-carbon compound. So keep track of the red. Those are the carbons that we started with. So 6 turns into 5. Remember, every time I strip a carbon off, I get CO2 and NADH. 5 goes to 4. Every time I take a carbon off, I get CO2 and NADH, right? So we can see the repetition there. So little by little, we are reducing that molecule with the help of NADH. So NADH is going to accept that we're reducing the NAD into NADH. So it's an electron carrier. Over here you see FAD is being converted into FADH2, and those are all going to take electrons into our electron transport chain, where we make lots and lots of energy, right? So this part is just talking about those electron carriers with NADH and FADH2. So the Krebs cycle will make lots of those, as you saw on the last slide. You can also see it here. 3 NADHs and an FADH2 for each turn. Okay, so every carbon becomes NADH. And these are going to go into the electron transport chain, which is something you've already read about. You've already practiced with photosynthesis. The hydrogen ion gradient, remember, is going to be allowed to diffuse through the hydrogen ion. Let me start. The hydrogen ions will diffuse through the ATP synthase, which, remember, is like a turbine. It will combine ADP with inorganic phosphates to make ATP. So that is our end goal. So at this point, we've taken our 2 pyruvates from glycolysis, and we have converted them into carbon dioxide, 4 NADHs, and 1 FADH2 per pyruvate. So that's going to be 8 NADHs and 2 FADH2s. Along the way, we've made 2 ATP molecules. Okay, so the whole purpose of the Krebs cycle is to create those electron carriers, the NADH and the FADH2. The big amount of energy happens in the electron transport chain, which means we have to shuttle electrons through the process into the electron transport chain, which we can call chemiosmosis. Okay, so we're taking off electrons, taking off hydrogens. The hydrogens will go into our gradient. As we saw on our last multiple choice questions, that isn't the only hydrogen available, right? But it is hydrogen that is available to add to that concentration gradient that already exists. Okay, so in our step Friday, we're going to talk about how NADH and FADH2 play a role. So very similar to electron transport or photosynthesis, but not identical. We're going to recognize some differences when we get into this on Friday. Okay. So I go through that quickly. You guys, in your review, in your study, how you're preparing, any of these processes you should draw out. Larger than life, sticky notes all over your wall, on a piece of paper, whatever you need to do. But you need to be able to draw these in order to be able to recall step. Thursday, I keep, so it was asked, are we Zooming Thursday or Friday? I just keep referring to our last day as Friday. I always mean Thursday. We won't ever meet other than Monday, Wednesday, and Thursday. So thank you for that clarification in the chat box. Okay, what questions do you have about the link reactions, glycolysis, or the Krebs cycle? Today we have a POGO covering these 2 processes, the link reactions, a little review of the glycolysis, link reactions, and the Krebs cycle. And I'm saving the electron transport chain entirely for Friday, so I'm not getting into that yet at all. Any questions about cellular respiration, AP biology, what's going on in the world?